were hybridized using end-labeled oligonucleotide probes complementary to the small RNAs. Haley et al. 2003. Tang and Zamor, 2004. The sequences of the small RNA probes are listed in Supplemental Table 2 online. The reverse primers, such as 165 to 166 STTMSWA8NT-link PR, 156 to 157 STTMSWA48NT-link PR, and D7D8 STTMSWA48NT-link PR, see Supplemental Table 1 online, were used as probes in RNA gel blots to detect STT-mRNAs from the transgenic plants. mRNA analysis by qRT-PCR. For examination of the mRNA levels of the genes of interest, total RNA was extracted using an RNeasy mini-kit, QIAGEN. Purified RNA was first treated with DNASI to remove any potential genomic DNA contamination and then used for RT with a high-capacity cDNA archive kit, Applied Biosystems. qRT-PCR was performed with an Applied Biosystems Step 1 instrument using the SYBR Green PCR Master Mix Kit, Applied Biosystems, according to the manufacturer's instructions. Actin mRNA was used as an internal control. The sequences of primers are listed in Supplemental Table 3 online. Values were obtained by normalizing to actin and then comparing the normalized values to those of control plants. The relative levels of gene expression were calculated using the 2-dd cycle threshold method. Livik and Schmidgen, 2001. 3 biological replicates were examined to ensure reproducibility. Myrna qRT-PCR. RT and the qRT-PCR for the quantification of miR-166 were performed according to Tachman's small RNA assays protocol. Applied Biosystems. The Tachman gene expression master mix, Applied Biosystems, was used for qRT-PCR. Arabidopsis SNO R101 was used as an internal control. The sequences of primers are listed in Supplemental Table 3 online. In vitro assay for miR-165-166 sequestration by STTM-165-166-48. In vitro production of STTM-165-166-48 RNAs was conducted by in vitro RNA transcription using T7 RNA polymerase and the DNA templates made by PCR with a pair of primers named T7 STTM-165, 166, 48P-F and T7 STTM-165, 166, 48P-R as well as a DNA oligo template named STTM Spacer 48NT template. See Supplemental Table 1 online. Transcription was conducted as described previously. Haley et al. 2003. Tang and Zamor, 2004. Synthetic miR-165-166 was produced in Dharmacon RNAi Technologies at Thermo Fisher Scientific. See Supplemental Table 2 online. Hot miR-165-166 and STTM-165-166 were made by 5 feet end labeling as previously described. Haley et al. 2003. Tang and Zamor, 2004. Annealing of STTM-165-166-48 with miR-165-166 was conducted in 2 times SSC. 1 times SSC is 0.15 m sodium chloride and 0.015 m sodium citrate. Buffer first by heating the RNAs at 95 degrees Celsius for 5 minutes and then by cooling the mixture to room temperature for an additional 2H of hybridization. The hybridized STTM-mRNA complex was first evaluated on native gel and then subjected to sequestering assay using 15% denatured page. The sequestering results were evaluated using a phosphor imager. Small RNA deep sequencing. RNA samples from the aerial parts of 3-week-old seedlings were isolated. Small RNAs were isolated from total RNAs following size selection using polyacrylamide, urea gel electrophoresis. Purified small RNAs were ligated to a 3' Celexa DNA adapter first. The 3' ligated product was then purified and ligated to a 5' Celexa RNA adapter. The small RNA library was reverse transcribed and then amplified by PCR. Small RNA sequencing was conducted using Genome Analyzer 2, Illumina. As previously described, Gildiel et al. 2008. Sequence data from this article can be found in the Arabidopsis Genome Initiative or GenBank, EMBL databases under the following accession numbers. miR-165-166, AT1G01183, AT4G00885, AT2G46685, AT3G61897, AT5G08712, AT5G08717, AT5G41905, AT5G43603, and AT5G63715, Mir 156 157, AT2G19425, AT2G25095, AT4G30972, AT4G31877, AT5G10945, AT5G11977, AT5G26147, AT5G55835, AT1G48742, AT1G66783, AT1G66795, and AT3G18217. Mir 160, AT2G39175, AT4G17788, and AT5G46845. Mir 168, AT4G19395 and AT5G45307. Tasserna D7, and D8, JA643511 and JA643515. PHB, AT2G34710, PHV, AT1G30490, REV, AT5G60690, ATHB8, AT4G32880, ATHB15, AT1G52150, IPS1, AT3G09922, ARF17, AT1G77850, and ACTIN2, AT3G18780. Supplemental data. The following materials are available in the online version of this article. Supplemental Figure 1. The optimal length of the spacer between the two small RNA binding sites in STTM. Supplemental Figure 2. The secondary structures and thermodynamic stabilities of various STTM 165, 166s with different spacer lengths. 8, 31, 48, 88, and 96 nucleotides. Supplemental Figure 3. Mutation in the spacer region changed the stem structure and reduced the efficacy in the functional blockage of Mir 165-166. Supplemental Figure 4. Comparison of the expression levels of Mir 165-166 in STTM 165-166 and MIM 165 transformants and vector control plants. Supplemental Figure 5. Comparison of the expression levels of Mir 165-166 targets in STTM 165-166 transformants with different spacer lengths. Supplemental Figure 6. Comparison of the expression levels of STTM and Mir 166 in STTM 165-166 del48, STTM 165-166 MUT48, STTM 165-165-48, and STTM 165-166-48 transformants. Supplemental Figure 7. Diagrams of the STTM-3148 plasmids and their vector control as well as the real-time primer locations. Supplemental Figure 8. Comparison of the phenotypes and the expression levels of the Mir 160 target, ARF17, in vector control, MIM 160, and STTM 160-160s-48. Supplemental Figure 9. Comparison of the expression levels of MIM, STTM, and Mir 165-166 in MIM 165 and STTM 165-166-48 transformants. Supplemental Figure 10. The expression levels of Mir 156 primary transcripts, PRI Mir 156A and PRI Mir 156B, as well as the mature Mir 156 in STTM 156-157-48. Supplemental Table 1. Primers used for plasmid construction in this study. Supplemental Table 2. Synthetic mRNAs and small RNA probes used in this study. Supplemental Table 3. Real-time PCR primers used in this study. This work was supported by the Kentucky Tobacco Research and Development Center and the USDA National Research Initiative Grants 2006, 35301, 17115 and 2006, 35100, 17433. National Science Foundation grants. MCB 0718029. Subaward S0000260 and iOS 10482216. An award from the Kentucky Science and Technology Corporation under contract KSTC 14440108029. Startup funds from Michigan Tech University to GT. And grants from the National Institutes of Health, National Institute of Diabetes and Digestive and Kidney Diseases. K01DK078648, 01 and R03DK084166, 01, to XT. Detlef Weigel and Jixin Xie kindly provided the MIM 165, MIM 160, and RDR6 to 15 mutant, respectively. Chengjian Li and Philip Zamore provided the training and guidance in small RNA library construction and the practice for small RNA deep sequencing at the University of Massachusetts Medical School. UTN and Zhuguo, Zhou. Zhou performed the bioinformatic analysis of the small RNA deep sequencing data. Author contributions. GT. Conceived and directed the project. GT and JY. Designed the research strategies. JY and YG. Conducted the majority of experiments. XJ. Conducted the in vitro assay for MIR 165-166 sequestration experiment. WK. Conducted some RNA gel blotting experiments. SP. Constructed some STTMs for their expression in Arabidopsis. XT. Conducted some data analysis and guided radioactive and real-time experiments. XC. Conceived the experiments with the SDN1-SDN2 double mutant. YG. Did the framework and JY. Conducted the deep work. Thus, both contributed equally to this article. GT, JY, and XC wrote the article. All authors contributed to writing the article. The author responsible for distribution of materials integral to the findings presented in this article in accordance with the policy described in the instructions for authors. www.plantcell.org. Is Juliang Tang, Jitang1 at mtu.edu. www.plantcell.org/cgi/doi/10.110/tpc.111.09444. 1. These authors contributed equally to this work. 2. Current address. Department of Horticulture and Landscape Architecture, Purdue University, West Lafayette, Indiana, 47907. C. Some figures in this article are displayed in color online but in black and white in the print edition. W. Online version contains web-only data. Received November 25, 2011. Revised January 18, 2012.